hybrid in a research field under ideal conditions. Later, they are cultivated in farmers' fields at different locations for 3 growing seasons. This helps in comparing the performance of the hybrid against the best crop cultivar available in that area. In this way, plant breeding has increased the yield of many crops, including wheat, rice, sugarcane, and millet. The Green Revolution is a result of using various plant breeding techniques to boost agriculture. During this period, there was a dramatic increase in the production of wheat from 11 to 75 million tons and rice from 35 to 89.5 million tons. The increase in production was credited to the semi-dwarf varieties of wheat and rice that were developed. The semi-dwarf wheat variety was first developed at the International Center for Wheat and Maize Improvement in Mexico by Nobel laureate Norman E. Borlaug. Likewise, the semi-dwarf rice variety, IR8, was developed at the International Rice Research Institute, IRRI, in the Philippines. Later, better-yielding varieties of wheat hybrids such as Sonalika and Kalyan Sona and rice hybrids such as Jaya and Ratna were developed in India. In the case of sugarcane, Saccharum barberi and Saccharum officinarum were crossbred to get a hybrid. The hybrid obtained had both the characters of its parents, such as a very thick stem with higher sugar content, and the ability to grow in northern parts of India. Similarly, high-yielding hybrids of millet, such as Jawar and Bajra, which are resistant to water scarcity, were also developed in India. In this manner, through various plant breeding techniques, many high-yielding crops were developed that helped boost Indian agriculture. Plant breeding is the skillful use of different plant varieties to produce a desired plant type, thereby improving crop yield. But in most cases, crop production is affected by several crop diseases caused by pathogens such as fungi, bacteria, and viruses. Blast of rice, red rot of sugarcane, and late blight of potato are examples of fungal plant diseases, while black rot of crucifers is a bacterial disease, and tobacco mosaic virus and turnip mosaic virus are viral diseases. In such cases, plant breeding can be used to develop crops possessing resistance to a particular pathogen, provided the causative pathogen and its mode of transmission is known. Resistance can be defined as the crop's ability to prevent a pathogen from attacking it, and is determined by the genetic makeup of a particular crop. The genetic makeup of crops can be altered using plant breeding techniques like conventional breeding and mutation breeding. Conventional breeding involves hybridization and artificial selection. It involves several steps that include collection of germplasm, selection of disease-resistant parents from germplasm, hybridization of the disease-resistant parents, selection of disease-resistant hybrids, and evaluation of the selected hybrids. There are many disease-resistant hybrid varieties of different crops developed through conventional breeding. Let's take a look at some of them. Himgiri, a hybrid variety of wheat, is resistant to fungal diseases such as leaf rust, stripe rust, and hill bunt. While Pusa shubra, a hybrid variety of cauliflower, is resistant to fungal diseases such as black rot and curl. There is Pusa komal, a hybrid variety of cowpea that is resistant to blight disease caused by a bacterium, while Pusa sadabahar, a hybrid variety of chili, is resistant to viral diseases such as tobacco mosaic virus and leaf curl. The only drawback of these disease-resistant wild relatives is that they are low-yielding. So, the disease-resistant genes from these wild relatives need to be transferred to high-yielding cultivars. As the disease-resistant genes are available in the same crop species, the transfer of disease-resistance genes between the crops is done by sexual hybridization. For example, the yellow mosaic virus-resistant variety of bhindi, Parbhani Kranti, is grown by the transfer of disease-resistant genes from its wild relatives. Although many disease-resistant hybrid varieties are produced through conventional breeding, the process is limited by the number of disease-resistant genes available in different crop varieties and their wild relatives. As a result, another plant breeding technique called mutation breeding is widely employed. Mutation causes changes in the base sequence within genes, resulting in a new character not found in the parental type and results in genetic variations. Mutation breeding involves the use of chemicals or gamma radiations to induce alterations in the genetic makeup of crops. The second generation of mutation-induced crops possess new characters that are absent in their parents. These plants are screened for disease-resistant genes and then artificially picked for breeding. An example of mutant breeding is seen in a moong bean variety that is resistant to the yellow mosaic virus and powdery mildew. Apart from pathogens, pests also reduce crop production. As a result, there is a need to develop crops that are resistant to pests as well by using plant breeding methods. These involve the same steps that are involved in the development of disease-resistant plants. Germplasm of cultivars or wild relatives are used as a source of pest-resistant genes. Insect resistance in crop plants can be attributed to the morphological, biochemical, or physiological features of a crop. For example, hairy leaves on cotton make it resistant to jacids, while hairy leaves on wheat make it resistant to cereal leaf beetle. Also, smooth leaves and the absence of nectar in some cotton varieties protect them from bollworms. Similarly, solid stems of wheat protect it from stem sawfly, whereas maize is protected from stem borers due to its high aspartate and low nitrogen and sugar content. There are quite a few insect-resistant hybrid varieties developed through plant breeding. Pusa Gaurav, a hybrid variety of rapeseed mustard, is resistant to aphids, while Pusa Sem 2 and Pusa Sem 3 are hybrid varieties of flat bean that are resistant to jacids, aphids, and fruit borers. And hybrid varieties of bhindi, Pusa Savani, and Pusa A4 are resistant to shoot and fruit borers. In this manner, plant breeding techniques help improve crop production, as well as prevent the use of bactericides and fungicides that can be harmful. They help develop crops that are resistant to pests and pathogens and can assist in boosting food production. Plant breeding plays a vital role in meeting the food demand of the increasing world population. While more than 840 million people around the world lack daily meals, it was reported that more than 3 billion people are fighting hidden hunger. Hidden hunger refers to a poor diet or malnutrition, that is, a deficiency of essential micronutrients, proteins, and vitamins. As poor people cannot afford fruit and vegetables that are rich in nutrients, they survive on a diet containing staple food crops such as wheat, rice, and maize. However, the level of vitamin A and micronutrients such as iron and zinc is very low in such crops, which makes them vulnerable to deficiency diseases that affect physical and mental health. To overcome this problem and promote public health, initiatives like biofortification were introduced. Biofortification is selective breeding of crops with higher nutritional value, such as proteins, vitamins, and minerals. For example, Atlas 66, a hybrid variety of wheat rich in protein, was bred with native wheat crops to fortify them. As a result, biofortification results in high-yielding crops fortified with specific nutrients. Another example of a biofortified staple crop is an iron-fortified rice variety that is 5 times richer in iron compared to its native variety. Similarly, maize hybrids containing double the quantity of amino acids compared to the other hybrids was developed. Did you know that the Indian Agricultural Research Institute, New Delhi, used biofortification to develop several fortified fruits and vegetables? Examples include vitamin A fortified carrot, spinach, and pumpkin, vitamin C fortified tomatoes, mustard, and bitter gourd, protein fortified beans and garden peas, and iron and calcium fortified spinach. Despite biofortification and several plant breeding techniques to boost food production, the demand for food was ever increasing. The shift to a meat diet further increased the demand for grains, as food grains are also used as animal feed. For example, a cow needs to be fed around 7 to 10 kilograms of corn to get around 1 kilogram of meat from it. For this reason, in addition to biofortified foods, scientists proposed the use of microbes as a food and feed supplement. These single-celled microbes are rich in proteins and are referred to as single-cell proteins or SCP. And so they are cultured to be used as a rich source of protein food. For example, spirulina a blue-green algae, is cultured for its high protein and mineral content, and it is also a source of vitamins, carbohydrates, and fats. It is used as a food supplement for human beings and as a feed supplement for animals. Moreover, it helps reduce environmental pollution, as it can grow on wastewater from potato processing plants, straw, molasses, animal wastes, and sewage. Another example is Methylophilus methylotrophus, a bacterium that can produce 25 tons of proteins. Nowadays, fungi, such as mushroom, are accepted as edible. Scientists believe that soon, microbes too will be accepted as a food supplement. Tissue culture is a method used for plant propagation in addition to other traditional plant breeding techniques. It was during the 1950s that scientists found that any part of a plant grown in nutrient media under sterile conditions can give rise to a whole new plant. This property of plant cells to give rise to a whole new plant is called totipotency. Tissue culture uses this property of plant cells for asexual plant propagation in an aseptic environment. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators such as auxins and cytokinins. The tissue culture technique involves several steps. The first step is excising a small part of plant tissue like the root, shoot, or leaves of a plant. Then placing the cutout part or explant in a suitable nutrient medium, followed by allowing the explants to multiply and form a group of tissues called callus. The callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Therefore, tissue culture is performed using a small part of a plant to develop many plants, and so is also called micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called somaclones. Micropropagation or tissue culture is employed in the large-scale production of tomato, banana, and apple. This technique can also be used to propagate high-yielding, disease-resistant varieties in a short span. Even if a plant is diseased, tissue culture can be used to get a healthy plant using the explant of the apical or axillary meristem. This is because the meristem is always free of disease and microbes. Culturing of meristems has been successfully carried out in plants such as banana, sugarcane, and potato. In addition to asexual propagation of plants, tissue culture is also used to mix two different species of plants that are otherwise sexually incompatible. Here, individual cells are isolated from each plant species and are placed in a culture medium. The cell wall gets digested in both cells, which aids fusion of the two protoplasts of different species, resulting in a hybrid protoplast. The hybrid protoplast is called a somatic hybrid, which gives rise to a whole new plant and may contain the desired characters of both species. This process of developing a hybrid plant using somatic cells is called somatic hybridization. It was employed while fusing the tomato and potato plants to obtain a hybrid plant. As a result, the hybrid plant resembled the tomato, but it didn't combine all the desired characters as expected. In this manner, tissue culture, somatic hybridization, and other plant breeding techniques provide a huge potential for manipulation of plants to produce new varieties, boost food production, and enhance crops.